Good to see you all back. Jerry here with Intel 420. Now, before we step into the deeper breakdown of Skunk Number One, I want to position the conversation the right way. This isn't a strain story, a nostalgia piece, or another recycled retelling of cannabis folklore. What we're doing here is treating Skunk Number One as an agricultural event, a moment where genetics, environment pressure, and necessity intersected and produced a line that shaped the entire trajectory of controlled environment cannabis. For a lot of people, the name skunk brings up disconnected memories, mixed cultural narratives, or assumptions shaped by the modern market. None of that helps you understand the actual cultivar or the forces that built it. So, before getting into chemistry, lineage, breeding influence, or modern market drift, we need to rebuild the frame from the ground up and put Skunk Number One back into the conditions that defined its emergence. Once you see what growers were working with and what problems they were trying to solve, the significance of the line becomes self-evident. Everything, the stability, the canopy behavior, the resin consistency, and the eventual global influence, traces back to those conditions. With that foundation set, we can begin at the only logical starting point, the environment that demanded a new kind of cannabis and the selective pressure that shaped Skunk Number One into something completely different from the material circulating at the time. Understanding Skunk Number One begins with understanding the pressure cooker conditions that shaped the earliest controlled cannabis cultivation in late 1970s California. This was a period when producing cannabis meant working against surveillance, unpredictable enforcement, shifting power usage expectations, and the risk of abrupt disruption from neighbors, landlords, or utilities. Growers were not dealing with thoughtfully designed cultivation facilities. They were improvising out of necessity. Basements were insulated unevenly, garages were converted with crude plastic sheeting stapled to rafters, and spare rooms were wired with extension cords run far beyond their rated loads. The environmental conditions inside these improvised spaces bore almost no resemblance to the climates that shaped the Afghan, Mexican, and Colombian seed stock circulating at the time. For the first time in cannabis history, the growing environment was being shaped more by legal pressure and the need for concealment than by geography or season. These indoor spaces produced a very specific set of challenges. Temperature swung dramatically because heat from the lamps accumulated faster than ventilation could clear it, especially when exhaust routes had to stay hidden from public view. Humidity fluctuated unpredictably because growers had little control over air exchange. Light patterns were consistent only to the extent the mechanical timers succeeded in cycling on schedule. Nutrient regimes were experimental and often unbalanced because liquid fertilizers were becoming commercially available for the first time, and growers were still learning how plants handled concentrated salt solutions in containerized root zones. These conditions created a new selection landscape, cultivars that thrived in well-ventilated outdoor fields or high-elevation terraces struggled under the claustrophobic realities of heat accumulation, stagnant air, and inconsistent irrigation practices. What growers needed at this moment was not mystery or legend. They needed plants that could complete a cycle in tight quarters where airflow was poor, radiant heat was unavoidable, and sudden environmental shifts were routine. Many imported seeds failed outright. Long flowering tropical lines grew aggressively upward and outward, colliding with ceilings and walls before their reproductive development ever stabilized. Other plants yellowed early because their nutritional needs had been shaped by soils and climates nothing like the concentrated nutrient systems being improvised indoors. Growers who had once relied on the forgiving nature of open air now had to develop an eye for the signals that plants sent under stress, and the earliest attempts at identifying workable individuals came from this constant exposure to plants that were collapsing for reasons unrelated to genetics and tied entirely to the artificial habitat around them. This period also marked the beginning of intentional observation under constraint. Individual growers began tracking which plants tolerated abrupt changes in temperature, which handled inconsistent irrigation, and which survived crowding without succumbing to fungal pressure. They were not yet thinking in formal breeding terms, but they were filtering populations against a completely new set of environmental triggers. A plant that could push through heat spikes, maintain functional transpiration in stagnant air, and shift into reproduction without relying on long, stable day-night transitions was not just more convenient, it was functionally essential. These selection pressures had no historical precedent in cannabis agriculture, and they began shaping the trajectory of cultivars grown indoors for decades to come. This is the context that produced the earliest foundations of what would become Skunk Number One. Not a mythical plant appearing out of nowhere, but a response to a hostile, artificial environment where only a narrow subset of traits allowed reliable completion of the cycle. The earliest selections that fed into Skunk Number One's development came from growers paying close attention to which individuals could endure these new conditions long enough to justify making seeds from them. Plants that tolerated instability better than others became the starting material for the next generation, not because of hype, but because they were the only ones reliably finishing under the constraints imposed by Prohibition-era cultivation. By the end of the decade, growers had unintentionally created the first consistent feedback loop between environment and trait persistence in modern cannabis history. They were no longer selecting for yield in open sunlight or for phenotypic expression across a long outdoor season. They were selecting for survival in enclosed, volatile spaces. This shift reshaped which traits were considered valuable and set the stage for more structured work that followed. When growers began saving seed from plants that completed cycles under the improvised conditions of the late 1970s, they were not yet working within a stabilized line. They were working with hybrid populations generated from Afghan, Mexican, and Colombian seed lots that entered the country through underground channels. These populations contained wide genetic variance because each regional source represented a heterogeneous pool shaped by local cultivation practices, open pollination, and the absence of tight genetic control. Early hybridization attempts did not begin with the idea of fixing traits. They began with the simple act of recombining plants that managed to reproduce reliably indoors. Those first crosses created populations where recessive and dominant alleles inherited from very different ecotypes began interacting in unpredictable ways. From a biological standpoint, this produced segregating generations, where structural, developmental, and aromatic traits appeared in a broad spread rather than clustering around a single predictable form. Some offspring inherited allele combinations that pushed them toward elongated structures typical of equatorial narrow-leaf backgrounds. Others exhibited compact growth and accelerated reproductive development associated with broadleaf highland ancestry. Many plants carried intermediate expressions because hybrid populations routinely express multi-locus interactions rather than simple, dominant recessive patterns. These interactions impacted measurable traits such as internodal distance, branching density, rate of apical elongation, calyx to bract ratio, and timing of floral initiation. During the earliest cycles, growers observed how these segregating generations behaved without any structured breeding theory, but their actions nonetheless produced real selection pressure. They kept seeds only from individuals whose developmental trajectories made sense within the cycle length they were trying to maintain. Plants that transitioned late into flower carried genetic combinations that did not suit the shortened operational time frame of indoor cultivation. As these individuals were removed from the pool, traits controlled by alleles responsible for delayed reproductive onset were gradually reduced in frequency. This is an example of selection removing alleles that regulate long flowering phases, which are typical in equatorial stock. Similarly, individuals that produced floral structures too loosely arranged for efficient harvest were often excluded. This indirectly removed certain combinations of alleles that influenced axillary bud formation, inflorescence density, and calyx stacking. When growers favored individuals displaying coherent inflorescence formation, they unintentionally selected for versions of developmental genes associated with compact floral architecture. Another expression that drew attention was the distribution of resin across calyx surfaces. Plants whose resin appeared early and formed in uniform patterns were favored because early resin visibility aligned with the operational need to determine which plants were worth keeping before committing full resources to the entire cycle. These early expressions corresponded with genetic factors controlling glandular trichome density, initiation timing, and the biochemical machinery responsible for synthesizing the resin components. Across repeated generations, allele frequencies began to shift in measurable ways, even if growers were not explicitly tracking genetic inheritance. Certain traits became more common because they increased the likelihood that a plant would be selected for seed production. Traits that hindered cycle completion or produced inconsistent developmental patterns were removed unintentionally through the elimination of individuals carrying those alleles. Over time, this filtering effect caused some traits to appear across multiple individuals in a generation, which signaled that certain allele combinations were being reinforced simply because they increased fitness under the constraints applied. As cycles progressed, growers noticed that the most desirable individuals did not emerge randomly. They emerged from subgroups that repeatedly produced offspring with more aligned developmental timing, more coherent floral structure, and more recognizable resin characteristics. This emergent clustering is a hallmark of selection operating on multigenic traits within a hybrid population. It allowed early cultivators to identify which parent combinations consistently generated offspring worth evaluating further. Once that consistency appeared, even in a broad hybrid population, selection could focus more precisely on reinforcing the allele combinations responsible for the recurring traits. By the time these early efforts accumulated into what would later be recognized as the foundation material for Skunk Number One, the genetic landscape had already undergone several rounds of unconscious but directional filtering. Instead of random hybrid offspring scattered across every possible expression, growers were beginning to see repeatable patterns. These patterns were not yet a stabilized line, but they indicated that the population had reached a stage where recombination was starting to produce recognizable structural, developmental, and resin-related traits more frequently than chance alone would predict. Once the developing population was moved into environments where cultivation parameters could be held constant across repeated cycles, expression patterns that were previously obscured by variable conditions became measurable. These controlled setups allowed traits to be documented using consistent light intensity, fixed day-night schedules, regulated irrigation volumes, and reproducible nutrient concentrations. Under these conditions, the population began to reveal a narrow developmental bandwidth that distinguished it from the unrefined hybrid material it originated from. This bandwidth appeared in quantifiable traits such as growth rate acceleration, photoperiodic response timing, and axial elongation patterns, each of which could be recorded across multiple generations without environmental noise masking the underlying genetic contribution. The early vegetative phase provided the first identifiable point of comparison. Plants exhibited a relatively uniform rate of leaf initiation and node formation when grown under identical photon densities. Measurements taken at set intervals showed reduced scatter in the number of nodes produced during a fixed duration of vegetative growth. This reduction in variance indicated that the alleles influencing early growth kinetics were reaching a more consistent distribution across individuals. The shift from broad variability to tighter clustering suggested that previous selection cycles had already removed many of the genetic combinations that produced extreme outliers. The transition into reproductive development showed another structured pattern. When photoperiod was shortened, the majority of individuals within this population initiated pre-flower structures within a narrow time frame relative to each other. This synchrony was not imposed by the environment. It reflected internal developmental programming that had become more uniform through earlier selection events. Because the environmental variables were held constant, any alignment in timing could be attributed directly to similarities in the genetic regulation of floral initiation. This made the population suitable for mapping flowering curves, allowing cultivators to track the period between photoperiod change, stretch phase elongation, and the deceleration of vertical growth. Vertical elongation itself provided additional quantifiable metrics. Measurements taken during the first 2 weeks after photoperiod change showed that individuals followed comparable elongation ratios relative to their pre-flower height. Instead of some plants doubling, while others barely extended, the population exhibited a consistent elongation coefficient, which is a measurable trait governed by growth regulator pathways and internode differentiation genetics. The uniformity of this coefficient across individuals demonstrated that earlier selection cycles and allele frequency shifts had consolidated the trait into a reliable expression domain. Irrigation and nutrient response trials deepened the characterization. Because the inputs were controlled precisely, researchers could measure how the population responded to incremental changes in nutrient strength. Under these tests, individuals expressed highly similar thresholds where excessive nitrogen, phosphorus, or potassium began producing early visual indicators of stress such as marginal chlorosis, leaf edge deformation, or altered transpiration behavior. The alignment of these physiological thresholds suggested that the biochemical machinery regulating nutrient assimilation, ion balance, and osmotic control was becoming consistent across the population. These are complex, multigenic traits, and their convergence provided strong evidence that the line had passed through enough selection cycles for major biochemical pathways to reflect similar allele combinations. Flowering progression offered another domain for expression mapping. When grown under constant temperature, humidity, and airflow, the timing of calyx enlargement, the density of glandular trichomes, and the rate of pistil senescence followed nearly identical sequences across individuals. Observations taken at fixed-day intervals demonstrated that flower set development, resin initiation, and maturation progressed on almost parallel timelines. Variance existed, but remained within narrow limits, reflecting that the trait clusters responsible for reproductive development had been reinforced consistently generation after generation. This reproducibility across multiple cycles established that the population had moved beyond a merely hybrid state and was now displaying line-like behavior in its developmental clocks. Because these controlled environments produced records across many replicated cycles, they offered a data set capable of demonstrating that the population expressed its traits reliably under repeatable inputs. These consistencies were not assumed. They were documented through measurable parameters such as node count, elongation ratios, floral onset timing, nutrient threshold curves, and maturation sequences. By charting these parameters across successive generations, cultivators determined that the population's developmental architecture had reached a stage where its expression could be predicted accurately before seed was even germinated. Once a population demonstrates reproducible developmental patterns, its resin chemistry can be evaluated as a biochemical system rather than a collection of unrelated sensory traits. In Skunk Number One, the biosynthetic machinery responsible for terpene and cannabinoid production operates through pathways that were shaped by the allele combinations fixed during earlier selection cycles. These pathways follow predictable sequences involving precursor generation, enzymatic conversion, and metabolite accumulation. Because chemical composition is the product of enzyme activity, substrate availability, and pathway regulation, the cultivar's chemical identity reflects biochemical stability rather than environmental coincidence. The monoterpene domain begins with the conversion of geranyl diphosphate into lineage characteristic volatiles. Analytical evaluations under controlled conditions show that Skunk Number One expresses a strong bias toward myrcene synthesis. This indicates consistently elevated activity of terpene synthase enzymes responsible for converting geranyl diphosphate into myrcene rather than diverting the precursor toward other monoterpenes such as osamine or linalool. The prevalence of myrcene establishes a heavy, low volatility anchor point that persists through drying and curing because myrcene evaporates more slowly than many other monoterpenes. Secondary monoterpenes, particularly limonene and alpha-pinene, appear in measurable and repeatable ratios relative to the myrcene baseline. Their presence indicates stable expression of limonene synthase and pinene synthase activity within specific quantitative ranges. In replicated analytical assays, these ratios show limited deviation when grown under identical conditions, which demonstrates that the line's biochemical regulation directs monoterpene pathways toward consistent outcomes. This is not a subjective sensory pattern, but a measurable biochemical signature confirmed by chromatography. The sesquiterpene domain follows the farnesyl diphosphate pathway. Skunk number 1 shows a reproducible presence of beta-caryophyllene and humulene synthesized from this precursor. These compounds are produced through separate sesquiterpene synthase enzymes with activity levels that remain stable across independent runs when environmental inputs remain constant. Their presence indicates that the biochemical pathways governing farnesyl diphosphate allocation operate through enzyme expression patterns maintained across the line. These sesquiterpenes contribute to the cultivar's broader chemical fingerprint and provide structural balance to the monoterpene-heavy upper profile. Cannabinoid synthesis offers another domain for analysis. The cultivar consistently expresses an active tetrahydrocannabinolic acid synthase pathway, resulting in dominant production of tetrahydrocannabinolic acid as the primary cannabinoid. Under replicated conditions, the ratios of minor cannabinoids, such as cannabigerolic acid, cannabichromenic acid, and trace cannabidiolic acid, remain within narrow statistical bands. This indicates that the upstream pathways converting geranyl pyrophosphate and olivotolic acid into cannabigerolic acid, followed by enzymatic conversion into tetrahydrocannabinolic acid, are operating under similar regulatory controls across individuals within the line. Such alignment suggests that earlier selection cycles consolidated the allele combinations that govern cannabinoid synthase strength and substrate channeling. Glandular trichome density adds another chemical dimension because resin production depends on both enzyme activity and physical resin gland formation. Microscopic analysis under controlled conditions reveals that skunk number 1 develops a predictable ratio of capitate-stalked glands relative to capitate sessile glands. This ratio influences not only total resin volume, but also terpene retention, because stalked glands have a higher capacity for volatile storage due to larger secretory cavities. The repeatability of this glandular architecture across multiple runs shows that the developmental genetics controlling trichome formation work in concert with the biochemical pathways producing the resin's chemical content. Chemical coherence becomes measurable when metabolites maintain quantitative proportions across multiple cycles. In skunk number 1, the relative percentages of myrcene, limonene, alpha-pinene, beta-caryophyllene, and humulene remain consistent enough to produce identifiable chromatographic patterns. These patterns do not depend on sensory interpretation, but on analyte detection thresholds and retention times. Because these results repeat across controlled trials, the cultivar's chemotype can be defined as a structured biochemical output rather than a variable product of environmental noise. The physical architecture of skunk number 1 can be examined at the level of tissue organization and meristem behavior, independent of its environmental history, genetic filtering, or chemical profile. The cultivar displays a predictable pattern of primary and secondary meristem differentiation, which can be documented through anatomical examination of vegetative and reproductive tissues. During early development, the shoot apical meristem consistently produces leaf node pairs with a structural alignment that maintains near uniform phyllotaxy across individuals. This alignment determines the eventual spacing between nodes and directly influences branch emergence and floral site distribution. Internode formation follows a morphogenetic pattern regulated by the expansion of cortical and pith tissues, mediated by the vascular cambium. When measured across individuals grown under uniform conditions, the length of internodal segments falls within a defined range that reflects the cultivar's inherent growth programming rather than environmental artifacts. This internodal predictability affects the total number of reproductive clusters the plant is capable of supporting, as each node provides a potential branching point and floral initiation site. Vascular architecture is another consistent anatomical domain. Cross-sectional examination of stem segments shows a reproducible arrangement of xylem and phloem tissues relative to the cortex. In skunk number 1, the proportion of lignified xylem to softer phloem tissues maintains a ratio that supports rigid stem development without excessive brittleness. This vascular ratio influences the plant's ability to sustain mechanical load as floral biomass accumulates during late reproductive growth. The stem's structural integrity prevents deformation under increasing flower mass, enabling the plant to maintain upright growth patterns without requiring external support structures. This trait contributes to predictable biomass distribution across branches. Leaf morphology reflects the cultivar's balanced anatomical design. Leaflets display a medium-thick mesophyll layer with distinct palisade and spongy parenchyma zones. The palisade layer is sufficiently dense to support efficient light absorption, while the spongy layer maintains adequate gas exchange capacity. This anatomical balance influences photosynthetic efficiency and contributes to consistent biomass accumulation throughout the vegetative phase. The vascular pattern within each leaflet shows a primary midrib with lateral veins radiating in a stable, repeating pattern, which supports efficient transport of water and metabolites into and out of the photosynthetic tissues. Reproductive architecture becomes evident once the plant enters floral development. Calyx formation begins with the differentiation of bract primordia from floral meristems located at the axils of branch nodes. The cultivar produces bracts with uniform dimensions across the plant, indicating stabilized regulation of bract cell expansion. These bracts form the structural foundation for glandular trichome placement. Although the biochemical content of trichomes is addressed elsewhere, their physical distribution can be analyzed independently. Skunk number 1 shows a high density of trichomes positioned along the abaxial and adaxial surfaces of bracts, with consistent spacing between glandular heads. This pattern results from predictable meristem activity that governs where trichomes initiate during floral development. Flower cluster density is determined by the rate at which bract primordia develop into mature calyx bract units. In this cultivar, the clustering occurs along short branch segments due to the stable internode length described earlier. The resulting floral architecture features compact clusters that maintain consistent spacing between calyx units. This structural pattern influences airflow pathways within the cluster, but is not dependent on environmental stress factors. It is a function of developmental programming that controls cell proliferation and tissue stacking. Biomass allocation across the plant is another anatomical characteristic that remains internally coherent. Dry weight analysis of representative samples reveals that the distribution of biomass between stems, leaves, and floral material follows a consistent percentage relationship when plants are grown under standardized inputs. This internal biomass ratio reflects how the cultivar partitions assimilates into vegetative versus reproductive structures. The cultivar dedicates a substantial proportion of assimilated carbon to reproductive tissues during the latter half of the flowering cycle, a trait associated with the efficiency of its vascular and meristematic systems. Finally, the symmetry of floral clusters can be evaluated. Skunk number 1 typically exhibits bilateral symmetry in bract placement around developing floral axes. This symmetry allows predictable spatial repetition, which is a sign of stabilized meristem programming. When examined across individuals, floral cluster form remains within a narrow morphological range, which indicates that the underlying developmental architecture is well integrated despite the complexity of multigenic control. Metabolic flux in skunk number 1 can be evaluated by measuring how the cultivar allocates carbon, nitrogen, and secondary metabolite precursors across different controlled cultivation systems. The primary comparison domains are sealed indoor environments, moderated input greenhouses, and controlled outdoor fields where input variables such as light intensity, vapor pressure deficit, and root zone temperature differ enough to alter resource distribution. These differences in resource distribution are driven by the interaction between photosynthetic rate respiration demand, and precursor channeling behavior within terpene and cannabinoid metabolic pathways. Under sealed indoor conditions, photon density and photoperiod remain constant, which produces a stable photosynthetic assimilation rate across the light cycle. Measurements of leaf-level gas exchange show that skunk number 1 maintains a consistent ratio of carbon dioxide assimilation to stomatal conductance, resulting in a predictable accumulation of carbon skeletons used in the mevalonate and methylerythritol phosphate pathways. Because these pathways supply the building blocks for both terpene and cannabinoid synthesis, the indoor metabolic profile reflects a steady-state flux where precursor pools do not fluctuate sharply. Enzyme activity involved in terpene synthesis remains proportional to available substrates throughout the photoperiod, resulting in uniform metabolite accumulation across the flowering phase. In greenhouse systems, the cultivar experiences fluctuations in photon density, diurnal temperature shifts, and variable vapor pressure deficit. These external changes alter metabolic flux by increasing the plant's reliance on dynamic resource reallocation. When photon density spikes under clear conditions, skunk number 1 increases carbon assimilation rates, leading to temporary elevation in glyceraldehyde, 3-phosphate, and pyruvate levels. These compounds feed directly into terpene and cannabinoid precursor pathways. However, because temperature and vapor pressure deficit also rise, respiration rates increase in parallel. Elevated respiration increases carbohydrate consumption in mitochondrial pathways, which reduces the proportion of carbon available for secondary metabolite synthesis. As a result, greenhouse-grown plants display transient peaks and valleys in terpene precursor availability, producing chromatographic profiles with slightly wider variance in total terpene quantities, even when ratios remain recognizable. Controlled outdoor systems introduce the greatest metabolic variability because photon density may exceed indoor and greenhouse maxima, and diurnal rhythm affects both stomatal behavior and carbon assimilation capacity. Under high-intensity full-spectrum sunlight, skunk number 1 exhibits elevated photosynthetic flux during peak light hours, increasing the production of triose phosphates that feed into primary and secondary metabolic pathways. When temperature rises significantly, the cultivar increases its transpiration rate to stabilize leaf temperature, altering ion transport across root and leaf tissues. Increased transpiration accelerates the uptake of potassium and magnesium ions, which play roles in enzymatic activation. These ion shifts influence the activity of terpene syntheses and cannabinoid syntheses indirectly by modulating intracellular pH and cofactor availability. Nighttime metabolic flux differs significantly among the 3 systems. Indoors, temperature and humidity remain stable, enabling respiration to proceed at a consistent rate. In a greenhouse, temperature drops at night, reducing respiratory demand but also slowing enzymatic turnover, which temporarily decreases the conversion rate of stored precursors. In outdoor cultivation, nighttime temperature drops may be more extreme. These drops slow substrate conversion rates, but preserve volatiles within resin glands by reducing evaporative pressure. Metabolite retention increases during cooler nighttime periods, resulting in marginally higher total terpene recovery when flowers are harvested at optimal maturity. Root zone temperature also influences metabolic flux. Indoors, root zones remain within narrow thermal ranges, enabling nutrient ion uptake patterns that follow consistent diffusion and active transport ratios. In greenhouses and outdoor systems, root zone temperatures fluctuate with ambient conditions. Cooler root zones slow nitrate reduction and ammonia assimilation, reducing nitrogen availability for amino acid synthesis. Because some terpene synthase enzymes rely on nitrogen-rich cofactors, cooler root zones can reduce total terpene accumulation even if ratios remain stable. Greenhouse and outdoor conditions also modify carbon allocation between vegetative and reproductive tissues. Elevated ultraviolet intensity in greenhouse and outdoor environments induces the cultivar to allocate more carbon into protective secondary metabolites. This increase in carbon allocation toward protective metabolites reduces biomass accumulation in other tissues and can increase the concentration of certain metabolites when measured per gram of dry weight. In contrast, indoor-grown specimens allocate carbon more evenly across vegetative and reproductive tissues, producing larger floral clusters with total terpene quantities distributed across greater biomass. Metabolic flux analysis shows that skunk number one's chemical output is not fixed but regulated by dynamic interactions among light intensity, temperature, vapor pressure deficit, root zone conditions, and diurnal respiration. The cultivar demonstrates consistent pathway usage across systems, but the rate at which precursors enter and exit those pathways differs. This affects total metabolite quantities without altering the fundamental qualitative chemotype. Post-harvest chemistry stability in skunk number one can be described by examining volatility, oxidation, isomerization, and degradation pathways that affect both terpene and cannabinoid compounds once biological activity ceases. The moment the plant is removed from active growth, enzymatic processes slow rapidly, and chemical changes become driven primarily by external conditions such as temperature, oxygen exposure, humidity, and ultraviolet radiation. Because skunk number one produces a distinct distribution of monoterpenes and sesquiterpenes, each class undergoes different stability patterns determined by molecular weight, vapor pressure, and susceptibility to oxidation. Monoterpenes such as myrcene, limonene, and alpha-pinene possess high vapor pressures relative to their sesquiterpene counterparts, making them the first to undergo evaporative loss. Under curing conditions where humidity remains between 50 and 60%, myrcene experiences a measurable decline within the first 7 to 10 days. This decline occurs because monoterpenes diffuse out of glandular resin heads when exposed to air flows created by periodic burping or passive gas exchange. Although the cultivar retains its chemotype identity during this period, the absolute quantity of monoterpene steadily decreases as the moisture gradient inside the floral material shifts. Higher curing temperatures accelerate this process, as increased thermal energy raises the kinetic activity of volatile molecules, allowing them to escape the resin matrix more rapidly. Sesquiterpenes, such as beta-caryophyllene and humulene, exhibit lower volatility due to their larger molecular structure and lower vapor pressures. As a result, they remain stable for longer periods during curing and storage. Chromatographic analyses conducted over 60-day intervals show that sesquiterpene concentrations decline at slower rates compared to monoterpenes, even when stored at the same temperature and humidity. However, these compounds are more vulnerable to oxidative reactions that occur when oxygen molecules interact with unsaturated bonds in the terpene structure. Oxidation converts these compounds into oxygenated derivatives, altering aroma properties and shifting chemical composition without evaporative loss. Oxidation rates increase when stored in environments with elevated oxygen levels or exposure to ultraviolet light. Cannabinoid stability follows a distinct degradation pathway. Tetrahydrocannabinolic acid converts into delta-9 tetrahydrocannabinol through decarboxylation when exposed to heat or prolonged storage. Over time, delta-9 tetrahydrocannabinol degrades into cannabinol through an oxidative pathway. This reaction proceeds more rapidly at higher temperatures or in storage environments with increased oxygen exposure. Under optimal controlled storage at temperatures between 50 and 60 degrees Fahrenheit and low oxygen exposure, the rate of delta-9 tetrahydrocannabinol conversion to cannabinol remains minimal over the first 60 days. However, in uncontrolled conditions, conversion rates increase significantly, producing measurable shifts in the cannabinoid profile. The integrity of glandular trichomes influences post-harvest stability by determining how effectively resin heads retain volatile contents. Capitate-stalked resin heads possess larger secretory cavities, but their structural walls are more susceptible to rupture during handling or drying. When resin heads fracture, volatile compounds diffuse more rapidly due to increased surface area exposure. Mechanical handling during trimming or insufficient drying can cause high rates of trichome rupture, accelerating terpene loss. In contrast, intact resin heads maintain their structural integrity and preserve volatiles by limiting gas exchange with the external environment. Moisture content also affects chemical stability by controlling water activity within the floral matrix. Excessive moisture above 65% water activity increases the mobility of volatile molecules and creates conditions conducive to microbial growth, which introduces oxidative enzymes that further degrade secondary metabolites. Low moisture content below 50% increases brittleness and can fracture resin heads, again accelerating volatile loss. The optimal moisture window for terpene retention lies between 55 and 60%, where internal water activity supports stable resin head structure without increasing degradation risk. Non-volatile compounds, including the majority of cannabinoids and certain long-chain terpenoids, exhibit significantly higher stability under proper storage conditions. Their larger molecular size and lower vapor pressure reduce evaporative tendencies. However, oxidative degradation still occurs at measurable rates when oxygen remains present in the headspace of storage containers. Removing oxygen through nitrogen flushing or vacuum sealing decreases degradation and preserves the chemical profile for extended periods. Ultraviolet radiation accelerates both terpene oxidation and cannabinoid breakdown by supplying energy sufficient to initiate photochemical reactions. These reactions cleave chemical bonds and produce oxidized derivatives, reducing both potency and aromatic fidelity. Storage in opaque, ultraviolet blocking containers minimizes this degradation pathway. The rate of photodegradation correlates directly with wavelength and intensity, making ultraviolet light particularly destructive compared to visible spectrum illumination. Temperature is the final major factor influencing post-harvest chemical stability. Higher temperatures increase reaction rates by accelerating molecular motion, enabling faster evaporation, oxidation, and decarboxylation. Lower temperatures slow these processes, but must remain above freezing to prevent moisture crystallization within plant tissues, which can rupture glandular structures. Population drift within modern Skunk Number One seed market recreations can be evaluated by examining how successive seed reproductions, commercial breeding practices, and the absence of a centralized genetic reference standard have reshaped the cultivar's population structure over more than 4 decades. Drift occurs when trait frequencies within a population change due to random sampling, selective decisions by breeders, or the introduction of external genetic material. Because most contemporary offerings originate from independently maintained breeding lines rather than a single, continuously managed source population, modern products labeled as Skunk Number One often represent divergent genetic groups. Genetic drift begins when an original population is reproduced through limited parent selections. If only a small number of parental plants are chosen for reproduction, the next generation inherits a subset of the original trait distribution. Over repeated generations, these reduced distributions can shift further as breeders continue selecting plants based on desired outcomes that may not match the original line's stabilized traits. The result is a progressive narrowing or redirection of trait frequencies. This effect is amplified when breeders reinforce selections across multiple cycles without external reference material to maintain alignment with the original cultivar. Founder effects contribute to divergence across seed companies. When a company begins its own version of Skunk Number One using a limited number of seeds or clones, the resulting line immediately diverges from all others derived from different founder plants. With each reproduction cycle, the founder bias strengthens. After numerous generations, 2 lines originating from different founders may retain only partial overlap in their trait distributions. These differences can manifest in growth parameters, developmental timing, or secondary metabolite expression, even when both lines continue to be marketed under the same cultivar name. Another source of drift is unintentional selection for traits that were not defining characteristics of the original line. Commercial breeders often prioritize high-yielding individuals, rapid finishing times, or specific expression traits aligned with market preferences. These traits may not correspond to the historical trait profile of Skunk Number One, but become fixed within the new breeding line due to repeated selection. Over time, these shifts create modern populations that differ significantly from the stabilized population originally developed more than 4 decades ago. Introgression is another factor affecting modern skunk number one recreations. Some seed market versions incorporate external genetics, either deliberately or incidentally. Deliberate introgression occurs when breeders cross skunk number one with other cultivars to adjust certain characteristics, then back-cross toward the original profile. Incidental introgression occurs when pollen contamination introduces unrelated genetic material. Both forms of introgression modify allele frequencies and reshape population structure, even when the resultant plants are back-crossed or selected to approximate the target profile. These modifications reduce alignment with the historical genetic architecture. Marker analysis from independent laboratories shows significant variance among modern seeds sold under the Skunk Number One label. Samples from different commercial sources display inconsistent clustering patterns when evaluated using genetic markers associated with ancestry components such as indica-leaning broadleaf and tropical narrowleaf lineages. Some modern versions cluster closely with their supposed Afghan-Mexican-Colombian ancestry pattern, while others drift toward unrelated hybrid backgrounds, suggesting that divergent breeding histories have produced inconsistent population structures. Phenotypic heterogeneity is another indicator of population drift. When seeds marketed as Skunk Number One produce broad variation in growth behavior across a given seed pack, it reflects inadequate stabilization of trait expression. Historically stabilized cultivars produce consistent structural and developmental outcomes across individuals. In modern seed market recreations, widened phenotypic ranges indicate that the line has become less uniform, often due to generations of unaligned breeding practices. This heterogeneity is observable across traits such as plant stature, internode spacing, flower cluster density, and developmental timing. Chemotype variability also reflects drift. Although the original line exhibited coherent terpene ratios and cannabinoid patterns, modern recreations show increased chemotypic spread when analyzed under controlled conditions. Some versions maintain a recognizable chemical architecture, while others exhibit reduced coherence due to shifts in enzyme expression patterns inherited from divergent lineages introduced intentionally or inadvertently. This variability indicates that the biochemical pathways responsible for terpene and cannabinoid production are no longer uniformly regulated across modern seed market versions. The absence of a centralized genetic or phenotypic reference standard allows drift to persist and accelerate. Without industry-wide criteria defining what constitutes Skunk Number One, each breeder's version becomes its own reference. This decentralization ensures that no consistent population structure exists in the modern market. As a result, the label Skunk Number One describes a category of lineages that may share partial ancestry but no longer represent a unified, stabilized cultivar. This is where we end. I feel this is a good point before we get to much deeper. Thank you for watching, and welcome to everyone joining for the first time. For those who come back video after video, I appreciate you just as much. My goal here is simple: to break down cannabis science in a way that's clear, accurate, and accessible so anyone who's curious can learn without getting lost in misinformation or outdated assumptions. I share this material because plant science can get complicated fast, and most people never get the opportunity to see how these systems actually work beneath the surface. If today's breakdown helped you understand the subject in a deeper or more structured way, then I'm doing what I set out to do. Everything on this channel is strictly for educational and informational purposes. Nothing here encourages the use, sale, or cultivation of cannabis where it is not legal to do so. Always follow your local, state, and tribal regulations and make informed, responsible decisions based on the laws in your area. Thank you again for being here and for taking the time to learn something real. I'll see you in the next one.